Transgenic plant. Transgenic plant. Transgenic plants are plants that have had their genome modified through genetic engineering techniques, either by addition of foreign genes or removal of certain detrimental genes. The aim is to introduce a new trait to the plant which doesn't occur naturally in the species. Method GM plant. First using gene gun, second using Agrobacterium tumefaciens. The gene gun method, also known as the microprojectile bombardment or biolistic method, is the most commonly used in the species like corn and rice. In this method, DNA is bound to the tiny particles of gold or tungsten, which is subsequently shot into plant tissue or single plant cells under high pressure using gun. The accelerated particles are penetrating both into the cell wall and membranes. The DNA separates from the coated matter and it integrates into the plant genome inside the nucleus. This method has been applied successfully for many crops, especially monocots like wheat or maize. Agrobacterium tumefaciens. It involves the use of soil-dwelling bacteria known as Agrobacterium tumefaciens. The piece of DNA that infects a plant is integrated into plant chromosome through a tumor-inducing plasmid. The TI plasmid can control the plant cellular machinery and use it to make many copies of its own bacterial DNA. The TI plasmid is a large circular DNA particle that replicate independently in the bacterial chromosome. The importance of this plasmid is that it contains region of transfer DNA where a researcher can insert a gene which can be transferred to a plant cell through a process known as prototip. Application of transgenic plant. The first application is resistant to biotic or abiotic stresses. The next application is to improve crop yield and nutritional value. Lastly, transgenic plants act as bioreactors for recombinant proteins. BT cotton. These products contain lepidopteran-active insecticidal proteins derived from the bacterium Bacillus thuringiensis and were targeted at bollworms like the tobacco budworm and the cotton bollworm that are difficult to control using conventional insecticides. Crops such as BT cotton have been genetically engineered to suit a particular environmental risk. In the case of BT cotton, strains of DNA from the bacterium Bacillus thuringiensis has been transplanted into the plant's DNA in order to make it immune to larvae of insects that eat the plant. It can achieve that as the BT gene is responsible for producing toxic proteins to the insect larvae, but are safe for humans and other mammals or birds. As a conclusion, GM plant has benefit to both agriculture producers and consumers. With better technology development and proper scientific education to give public confidence, genetic engineering will become a powerful tool to our society to further increase agriculture production, improve nutritional values, and reduce environmental pollution. That's all for me. Thanks for watching.